Hey, welcome back! I'm Mr. Lewis, and today we're going to work through some Cambridge-style sample questions. As we go, I'll help clarify some of the questions that came up in class. If anything is still unclear, feel free to reach out. Let's start with question 1. We'll go step by step so you clearly see why these answers work. Part A. From the study by Pozzulo. In the actual study, the researchers deliberately chose two very different types of targets. The children watched short video clips of popular cartoons they knew well, Dora the Explorer and Go Diego Go, and video clips of unfamiliar adult human faces. The results were striking. Children correctly identified the familiar cartoon targets almost 100% of the time, but their correct identification rate dropped to only 23% for the unfamiliar human faces. So, target familiarity is the key factor that made identification easy or difficult. It directly influenced how well the children could recognize the person they had just seen. However, other answers like target present or target absent could be valid as a correct answer. Part B. Now that we understand the factor, let's look at how the identification was actually tested. Here, the key term is simultaneous lineup. After watching each video, the child or adult saw the entire lineup, either the target plus 3 foils, or 4 foils if the target was absent, all displayed together on the screen at once, plus a silhouette box for "not here." This is the classic definition of a simultaneous lineup. Everything is visible at the same moment, so the participant can look back and forth and compare the photos directly before making a decision. That's exactly what the study did. Part C. So now, let's connect this to what is happening inside the mind. This study relates to the cognitive approach because it examines memory and recognition processes. Let me explain this properly. The cognitive approach is all about what happens inside our heads, the internal mental processes we cannot see directly. The textbook describes memory as a 3-stage cognitive process: input/encoding, taking in information. In this study, watching the short video clip of the target. Processing/storage, holding that information in memory. Output/retrieval, bringing the information back out. In this case, recognition, comparing the stored memory of the target with the photos in the lineup. Quick note: in a lineup, the foils are the incorrect people shown with the target. They look similar but are not the correct answer. The children first encoded the target from the video. Later, they had to recognize it in the lineup by mentally comparing their memory with each photo. The researchers made the task easier or harder on purpose. With familiar cartoons, encoding and recognition were straightforward, so children got almost 100% correct. With unfamiliar human faces, encoding was weaker and recognition was much harder, so accuracy dropped to 23%. The study shows that identification accuracy is not just about what the child saw. It depends on how well the information was encoded, stored, and retrieved. Let's go through question 2 together, part A. Your answer is: the sample consisted of 40 males aged 20 to 50 years. Participants were drawn from New Haven and the surrounding communities and volunteered after seeing a newspaper advertisement. They responded to a newspaper advertisement and direct mail solicitation because they thought they were participating in a study of memory and learning at Yale University. He even includes a table showing the age groups and occupations, which could be blue or white collars: postal clerks, teachers, salesmen, engineers, laborers, etc. Part B. Now, moving from the sample to what they actually found. In the results section, Milgram reports the key finding. Although the learner was screaming, pounding on the wall, and eventually stopped responding, 26 of the 40 ordinary men kept going all the way to the highest shock level labeled danger severe shock, and even beyond led the XXX switches. In the discussion, Milgram emphasizes how surprising this was. People had learned from childhood not to hurt others, yet the majority obeyed the authority figure completely. This is the main conclusion of the whole study. Ordinary people can be led to commit harmful acts when an authority tells them to. So here, behavior is influenced by authority. Next, we shift to how behavior changes depending on social relationships and distance. Let's look at question 3 on Perry et al. Personal space. Question 3, Part A. The paper describes 2 clear behavioral measures. In experiment 1, they measured approach distance using the comfortable interpersonal distance (CID) task, and in experiment 2, they measured seating distance, chair distance in the room photographs. Part B. Now, let me walk you through how personal space was tested in this study. The researchers used 2 computer-based experiments to measure how comfortable people felt at different distances. Experiment 1. Comfortable interpersonal distance, CID. In the first experiment, participants watched short animations of a virtual room. A figure entered the room from one of 8 doors and then moved toward them. This figure could be a stranger, a friend, an authority figure, or even a ball used as a control condition. Participants had to press the space bar to stop the figure at the point where they felt comfortable. The distance was measured as a percentage. So 0% means the figure was very close, almost touching. 100% means they stopped it immediately. So these gave a very precise and objective measure of personal space. Experiment 2. Room selection task. In the second experiment, participants were shown pairs of room images. The only difference between the images was the distance between 2 chairs, which ranged from 20 centimeters to 140 centimeters. Participants had to choose the room they preferred for an imagined conversation. There was also a control condition, where they judged distances between objects, like a table and a plant, and not people. Quick note about the design. Both experiments used a double-blind design, meaning neither the participants nor the researchers knew who received oxytocin and who received a placebo. Participants were also divided into high empathy and low empathy groups to see how personality affects personal space. Part C. Now, based on that setup, let's look at the results. In the results section, the researchers report a significant main effect for condition. Bonferroni corrected pairwise comparisons showed that the mean percentage of maximum distance was much smaller when the approaching figure was labeled friend, 12.46%, compared with a stranger, 39.82%, or authority figure, 34.12%. This result relates to same-sex versus opposite-sex distances because the study used only male participants and only male animated protagonists. The method section states that all 54 participants were healthy males, and the protagonists in the comfortable interpersonal distance (CID) task and room selection task were all male figures. Therefore, all the interactions observed were same-sex hot male-male interactions. The paper itself notes this as a limitation in the discussion. Because the sample was all male, the researchers cannot conclude whether the same pattern closer distances to friends would occur in female participants or in mixed-sex, opposite-sex interactions. This means the study provides clear evidence of how familiarity affects preferred distance within same-sex male pairs, but we do not know if opposite-sex distances would show the same or different pattern. So far, we have looked at social behavior. Now we move into a clinical case, how fear develops and is treated. Let's look at question 4 on Saavedra and Silverman's button phobia case study. Part A. One symptom of the boy's phobic response was avoidance of buttons, or high levels of subjective distress/disgust when buttons touched him/crying and distress when buttons fell on him. In the case report, the researchers describe how the 5-year-old boy developed an intense fear of buttons after a traumatic incident at age 5. From that point, he showed clear avoidance behavior. He refused to wear any clothes with buttons, avoided touching buttons, and would have tantrums or cry if buttons came near him or touched his skin. The paper also reports that on the feelings thermometer, he gave buttons the maximum distress rating of 8 out of 8 and displayed strong disgust reactions. This symptom is not just everyday dislike. It directly meets the DSM-4 diagnostic criteria for specific phobia that the researchers used in the study via the ADISC/P interview. According to DSM-4, a specific phobia involves a marked and persistent fear that is excessive or unreasonable and is skewed by the presence or anticipation of a specific object, in this case, buttons. Exposure to the phobic stimulus almost invariably provokes an immediate anxiety response: crying, tantrums, high distress ratings, disgust. The situation is avoided or endured with intense distress, and the avoidance or distress causes significant interference with normal routine. For example, he could not dress normally or attend school comfortably. The study explicitly states that the boy met full DSM-4 criteria for specific phobia of buttons. Mentioning DSM-4 here is useful because even though we currently use DSM-5-TR, the original 2002 paper diagnosed the case using DSM-4. It shows you understand exactly why the researchers identified these behaviors as clinical symptoms of a phobia rather than ordinary fear. Part B. One step in the treatment to reduce fear was that when in vivo, behavioral exposure alone increased the boy's distress ratings, the therapist added disgust-related imagery exposures, imagining buttons falling on him or touching his body, combined with cognitive restructuring. Let me explain exactly what this step involved and why it was important, straight from the original paper. The researchers used a graduated exposure hierarchy, Table 1 in the paper, based on the boy's own disgust/fear ratings, 0 to 8 on the feelings thermometer. They began with a standard in vivo, real-life exposure, starting with low anxiety items such as a large denim button held at a distance, then gradually moving closer until he could touch it. However, as treatment progressed, the boy's distress ratings on some hierarchy items actually increased despite behavioral improvement. This is clearly shown in Figure 1. The therapist realized that the phobia had a strong disgust component, evaluative learning, rather than just fear. They therefore added a new step: disgust-related imagery exposures. In these sessions, the therapist guided the boy through vivid imaginary scenarios, such as imagine buttons falling onto your skin, or imagine buttons touching your body and clothes. While he was imagining these scenes, they paired the imagery with cognitive restructuring, helping him challenge and replace his catastrophic thoughts For example, buttons are disgusting and dangerous. With more realistic ones, for example, buttons are harmless pieces of plastic/metal. This combination was repeated until his distress ratings dropped significantly, often from 8 down to 3 or lower. Only then did they return to the in vivo exposure tasks. The paper reports that this switch was crucial. It targeted the disgust emotion that pure behavioral exposure could not fully address, allowing the boy to complete the full hierarchy and achieve complete remission of the phobia at 6 and 12-month follow-up. This step demonstrates the study's strength in adapting treatment in real time based on the boy's ongoing data, which is a key advantage of the case study method. Part C. The paper highlights that because it was a single case, they could watch the boy's reactions in real time. Figures 1 to 3 and change the treatment when standard exposure actually made his distress ratings go up. This personalized idiographic approach led to complete remission that lasted at 6 and 12-month follow-up. It's a clear explanation that is fully applied to this study. Exactly what gets the full 2 marks. These answers are short enough to write quickly in the exam, but still complete and accurate. From a single clinical case, we now move to biological explanations of behavior. Question 5 from Hassett et al.'s study on monkey toy preferences. A. One type of toy used in this study was wheel toys. In the method section of the paper, the researchers specifically presented the monkeys with 2 categories of human toys. Wheel toys, such as wagons, trucks, cars, construction vehicles, and plush toys, such as teddy bears, Winnie the Pooh, Raggedy Ann. They chose these 2 distinct types to see whether monkeys would show the same sex type preferences as human children. So wheel toys, or plush toys, is a correct type used in the study. B. One finding related to sex type toy preference was that male monkeys showed a strong preference for wheel toys over plush toys, while female monkeys showed no significant preference. This comes directly from the results section. The researchers found a significant interaction between sex and toy type. Male rhesus monkeys interacted significantly more with wheeled toys than with plush toys, demonstrating a clear and consistent preference. Female monkeys, in contrast, interacted with both types of toys more equally and showed no statistically reliable preference. Their behavior was more variable. This pattern closely mirrors the sex differences seen in human children. C. One way this study fits the biological approach is that it demonstrates sex differences in toy preferences in rhesus monkeys without any gendered socialization, suggesting the preferences have a biological basis. Let me explain this in detail. The biological approach emphasizes that some behaviors are influenced by innate biological factors, such as hormones and evolutionary predispositions, rather than being learned purely through culture or socialization. In this study, the researchers tested rhesus monkeys living in a natural social group at the Yerkes Primate Center. Monkeys are not exposed to human-style gender socialization. There are no boys' toys or girls' toys messages in their environment. Despite this, the male monkeys showed the same strong preference for wheeled toys, which facilitate active manipulative play that is typically seen in human boys, while female monkeys showed the more variable pattern seen in human girls. The authors conclude that these preferences likely reflect hormonally influenced behavioral and cognitive biases that evolved to support different activity preferences. Because the study rules out social learning as the main cause, it provides strong evidence for the biological approach by showing that sex-type toy preferences can emerge from innate biological mechanisms. Now, from biological explanations, we move to learned behavior. Question 6. Here's my answer for question 6 from Bandura et al.'s aggression study. A. 2 control measures used in this study were: all children were given the same toys in the test room. All subjects underwent the same mild aggression arousal procedure before the test phase. The paper explains that every child, whether in the aggressive model, non-aggressive model, or control group, was tested in the exact same experimental room with the identical set of toys arranged in a fixed order. This ensured that any differences in aggressive behavior were due to the model they had seen, not to differences in the toys available. Similarly, before the imitation test, every child was taken to an anteroom with attractive toys and then told they could not play with them. This standardized mild frustration, aggression arousal, was used for all 72 children, so that differences in aggression during the test phase could not be explained by some children being more frustrated than others. B. One difference found between boys and girls in aggressive behavior was that boys displayed more physical aggression than girls. The results section clearly shows this. Boys who saw the aggressive model imitated far more physical acts, punching, kicking, hitting the Bobo doll with the mallet, than girls did. The paper notes that aggression is a highly masculine-typed behavior, and the data confirm that boys were more predisposed to imitate physical aggression, especially when the model was male. C. One weakness of the sampling method used in this study is that the sample consisted of 72 children from only one nursery school, Stanford University Nursery School. The method section states that all the boys and girls were enrolled in the Stanford University Nursery School. Because they came from the same single nursery, the sample had limited socioeconomic and cultural diversity. This means the results may not generalize well to children from other backgrounds, different social classes, or other parts of the country. The study therefore has reduced external validity because we cannot be sure the same patterns of imitation would appear in a more diverse group of children. Question 7 from Hölzel et al.'s study on mindfulness and brain scans. A. One brain region measured in this study was the left hippocampus. In the results section of the paper, the researchers carried out a region of interest analysis focused on the hippocampus and insula because previous cross-sectional studies had shown structural differences in meditators in these areas. They used voxel-based morphometry on the anatomical MRI scans and found a significant increase in gray matter concentration, specifically in the left hippocampus after the mindfulness training. BB. One way MRI scanning was used in this study was to measure gray matter concentration before and after the mindfulness training. The method section explains that high-resolution anatomical MRI images, T1 weighted image sequence, were collected from all participants twice. Once in the 2 weeks before the 8-week mindfulness-based stress reduction (MBSR) program and once immediately afterwards. The same scanning procedure was used for the waitlist control group approximately 2 months apart. These MRI scans were then analyzed using voxel-based morphometry (VBM) to quantify changes in regional gray matter density. This allowed the researchers to detect structural brain changes that occurred as a direct result of the mindfulness intervention. C. One finding from this study in relation to changes in brain structure was that participants who completed the MBSR program showed a significant increase in gray matter concentration in the left hippocampus compared with the waitlist control group. Let me explain this in detail. The hippocampus is known to be critically involved in learning and memory processes and in the modulation of emotional responses and stress regulation. Therefore, the authors conclude that the observed increase in gray matter concentration in the left hippocampus provides objective evidence of structural neuroplasticity that may underlie the improvements in mindfulness, emotion regulation, and stress reduction that participants experience after completing the MBSR program. Now back to real-world behavior in natural settings. Question 8 from Piliavin et al.'s study on subway Samaritans. A. One characteristic of the victim used in this study was that he carried a black cane. In the method section of the original paper, the researchers described the 4 male victims, aged 26 to 35, who took part. On 65 trials, they appeared sober and carried a black cane, the cane or ill condition. On the other 38 trials, they smelled of liquor and carried a liquor bottle wrapped tightly in a brown paper bag, the drunk condition. The cane was deliberately chosen as a visible characteristic so that passengers would perceive the victim as ill rather than responsible for his own plight. This characteristic was therefore a key independent variable in the study. B. One piece of quantitative data gathered in this study was that the cane, ill victim, received spontaneous help on 62 out of 65 trials. The results section and Table 1 reports the exact frequencies of helping behavior. Spontaneous help, that is before any programmed model could intervene, occurred on 62 of the 65 cane trials, a helping rate of approximately 95%. In contrast, the drunk victim received spontaneous help on only 19 of 38 trials. This quantitative data clearly shows the powerful effect of the type of victim variable on helping behavior and was recorded by the 2 observers on every trial. Or C. One weakness of conducting this study in a field setting is that there was lack of control over extraneous variables such as crowding and noise inside the subway car. In the method section, the researchers report that the mean number of people in the car was 43, and the mean number in the critical area was 8.5. But these numbers varied widely, range 15 to 120 in the car. Because it was a real subway train, they could not standardize or control how many passengers were present, where they sat, or whether they moved out of the critical area. The paper also notes that people sometimes left the critical area when help was delayed. These uncontrolled extraneous variables, especially crowding, could have influenced how quickly or how many people helped, so we cannot be completely sure that the differences in helping were caused only by the type of victim or the presence of the model. This reduces the internal validity of the study compared with a laboratory experiment where the number of bystanders could have been held constant. Next, we look at learning in animals. Question 9 from Fagen et al.'s study on elephant learning. A. 2 behaviors recorded by the researchers were: trunk here. The elephant gently placed the distal end of her trunk on the trainer's outstretched palm. Trunk up. The elephant lifted her trunk upwards so that fluid could run to the base of the trunk. In the method and results sections of the original paper, the researchers describe how they broke the trunk wash procedure into separate trainable behaviors. They specifically recorded whether the elephant performed trunk here, placing the ventral aspect of the trunk tip gently on the trainer's palm, and trunk up, lifting the trunk in a curl or straight position. These two behaviors were shaped using lure and shaping techniques, and the researchers measured success rates and number of offers for each one during performance tests. Both behaviors had to be performed reliably before the elephants could move on to the full trunk wash chain. B. Two differences between Fagen et al. Les's study and Perry et al. Les's personal space study are: Difference 1. Sampling of participants. This is the one the question says must be included. Point. Fagen et al. Used a small naturalistic sample of 5 female working elephants, whereas Perry et al. Used a larger sample of 54 male human volunteers. Evidence: Fagen et al. Selected 5 female elephants, 4 juveniles aged 5 to 7 and 1 adult, from the same stable in Nepal, chosen by staff for docility and handler willingness. Perry et al. Recruited 54 healthy male volunteers and split them into high and low empathy groups using the Interpersonal Reactivity Index. Explanation: this sampling difference means Fagen et al.'s study has higher ecological validity because the elephants were studied in their everyday captive conditions. However, the very small, non-random, all-female sample limits how far we can generalize the results. In contrast, Perry et al.'s larger screened human sample allows clearer comparisons between empathy levels and has more statistical power. Link/comparison. Overall, Fagen et al.'s sampling is stronger for understanding real animal behavior in a natural setting, while Perry et al.'s sampling is stronger for controlled human comparisons. Difference 2. Method and setting point. Fagen et al. Carried out the study in the elephant's real stable environment using only positive reinforcement, whereas Perry et al. Used a highly controlled laboratory setting with computerized tasks and double-blind drug administration. Evidence: Fagen et al. Trained the elephants while they were leg-chained in their usual stalls, recording minutes of training, number of offers, and success rates across short sessions. Perry et al. Tested participants in a virtual room, comfortable interpersonal distance, CID task and room selection task under double-blind oxytocin/placebo conditions with a waitlist control group. Explanation: Fagen et al.'s field-style approach therefore has higher ecological validity because the behaviors occurred in a real-life context, but it has lower internal validity because of uncontrolled variables such as Mahout presence and daily routines. Perry et al.'s lab design allows precise cause and effect conclusions about oxytocin and empathy, but in a highly artificial situation. Link/comparison. In summary, Fagen et al.'s method is stronger for real-world applicability, while Perry et al.'s method is stronger for scientific control and replicability. Finally, we finish with one of the classic studies on sleep and dreaming. Question 10. Here is my evaluation of the study by Dement and Kleitman, 1957, on sleep and dreams. Strength 1 is that the study collected a large amount of quantitative data showing a clear link between REM sleep and dreaming. Across 351 awakenings on 61 nights, participants recalled dreams after 152 out of 191 REM awakenings, about 80%, but only after 11 out of 160 NREM awakenings, about 7%. This quantitative data is a strength because it provides objective, measurable evidence of the relationship between REM periods and dreaming, rather than relying only on subjective reports. It makes the findings much more reliable and scientific. Strength 2 is that the researchers used objective physiological measures, including EEG recordings and eye movement recording. They attached electrodes near the eyes and on the scalp to continuously monitor brain waves and rapid eye movements throughout the night. This is a strength because it allowed them to identify REM periods accurately and independently of what participants said after waking, reducing bias and making the link between REM sleep and dreaming more objective and replicable. Weakness 1 is that the sample was very small and unrepresentative. Only 9 participants took part in the study, and 7 of them were male. This is a weakness because the results may not generalize to women, children, or the wider population, limiting how far we can apply the findings. Weakness 2 is that the artificial laboratory setting lacked ecological validity. Participants had to sleep in a quiet lab room with electrodes attached to their heads and eyes, and they were awakened repeatedly by a loud doorbell. This is a weakness because the unnatural environment and frequent disturbances may have altered normal sleep patterns and dream content, so the results may not fully reflect everyday dreaming that occurs naturally at home. Overall, while the study had important strengths in its use of quantitative data and objective measures, its small sample and artificial setting limit the generalizability and ecological validity of the conclusions. Alright, that's everything for today. If you notice, all of these answers follow the same idea: clear knowledge, direct reference to the study, and simple explanation. That's exactly what examiners are looking for. Now your job is to try these again on your own, because that's where the real learning happens. And if anything is still unclear, just reach out. See you in the next class.